Welcome to the Sans Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Please hit that like button and subscribe right now. It truly helps support the Sans Science Experience and allows us to keep bringing you these deep dives into fascinating scientific discoveries. Today, we're diving into something, well, fundamental that you might think you know pretty well, but it turns out it's far more intricate than maybe we first imagined. Mitochondria.
Yeah.
For years, we primarily saw them as the cell's powerhouses, right? Just churning out energy ATP. But our source material for this deep dive, a fantastic review from Samuel Weinberg and Navdeep Chandal, reveals a revolutionary shift in how we understand these tiny organelles.
That's absolutely right. This deep dive is really all about uncovering how mitochondria, specifically through something called mitochondrial reactive oxygen species, or mtROS, how they act as critical signaling hubs. They're actually orchestrating your immune cell function, guiding metabolic adaptation, and even playing a vital role in defending against pathogens.
And ROS reactive oxygen species? Most people probably associate that with damage, right? Oxidative stress.
Exactly. You might primarily think of ROS in terms of damage, but we're going to unpack how, in controlled amounts, they're absolutely essential messengers for your body's complex defense systems. We'll explore their intricate dance in both your immediate innate immunity and your more specific adaptive immune responses. It really changes the game.
Okay, let's unpack this then. Our source makes it clear that mtROS, once seen mainly as these harmful byproducts, are now understood as indispensable signaling molecules. So what exactly are they, these mtROS, and where do they come from inside the mitochondria?
Well, at its core, mtROS largely comes from the mitochondrial electron transport chain, the energy production line, specifically from complex 1 and complex 3. That's where electrons can sometimes incompletely reduce oxygen, forming superoxide.
Superoxide.
And then the superoxide, it's very quickly converted into hydrogen peroxide, H2O2, by enzymes like SOD1 and SOD2.
And what's really striking here is that their generation isn't just constant. It's highly dynamic, you said, almost like a dimmer switch.
Precisely. It's very dynamic. When your cells need a lot of energy, a lot of ATP, the mitochondrial membrane potential is lower. That reduces electron leakage and basically limits mtROS production.
Less leakage, less ROS. Makes sense.
But when ATP demand is low, that membrane potential builds up. It gets higher. And that increases the chance for electrons to leak out, leading to elevated mtROS, particularly complex 1 and complex 3.
Okay, so the cell's energy state directly controls this ROS signaling level. And this is where it gets really interesting. This idea of reverse electron transport, or RET.
Yes, RET, it's a key mechanism, especially complex 1. It's often linked to a molecule called succinate. Essentially, electrons can be pushed backward from a highly reduced coenzyme Q pool back into complex 1, and that generates superoxide right there in the mitochondrial matrix.
Push backward, okay.
Yeah, against the usual flow. Meanwhile, complex 3, it produces superoxide on both sides of the inner membrane. And the superoxide produced on the outer side can then travel out to the rest of the cell, the cytosol, through channels like VDAC.
So we have ROS being made inside, and ROS that can signal outside the mitochondrion. What does this all mean for signaling, then? How does H2O2 hydrogen peroxide, sounds like something you put on a cut, how does that act as a messenger inside a cell?
Well, H2O2 is a critical signaling molecule because it can directly modify proteins. Specifically, it targets particular cysteine residues on proteins. Think of it like adding a small chemical tag. This tag changes the protein's function.
So it's specific, not just random damage.
Exactly. And you have enzymes like Peroxiredoxins, PRDXs, and Glutathione peroxidases, GPXs. They're not just antioxidants cleaning up ROS. They act as redox sensors. They control H2O2 levels, sure, but they also facilitate its selective transfer, its message, to other specific signaling proteins. It's called a redox relay.
A redox relay, like passing a baton.
Precisely. It ensures the signal goes to the right place.
But, okay, too much ROS is still bad, right? You mentioned oxidative stress, and our sources talk about something called ferroptosis.
Absolutely. Balance is key. While these optimal levels are crucial for signaling, if it gets out of control, dysregulation can lead to damage. Ferroptosis is a really good example. It's a specific type of cell death driven by iron-dependent lipid peroxidation. Basically, highly reactive hydroxyl radicals attack fats in cell membranes.
Ouch.
Yeah, it causes membranes to break down. But your immune cells have multiple antioxidant systems, like the GSH-GPX4 pathway and the NRF2 pathway to counteract this. They maintain this delicate balance. And we're also learning about redox microdomains, specific little areas in the cell where H2O2 signaling happens safely while other areas are protected.
Fascinating, like little communication hubs within the cell. Okay, let's dive into innate immunity, the first line of defense. Our source talks about how mtROS influences macrophages, those big eater cells of the immune system.
Right, macrophages are a prime example. An early link was found with certain adapters like TRFs and toll-like receptor signaling. Basically, mtROS production influences key inflammatory pathways like NF key activation and the production of pro-inflammatory cytokines. But what's really fascinating here, as we touched on, is that complex 1 and complex 3 mTROS seem to have distinct roles.
So it really matters where it comes from within the mitochondria, not just ROS is ROS.
Exactly. It's much more nuanced. For instance, complex 3-derived mTROS, specifically, seems essential for producing the anti-inflammatory cytokine IL-10. Studies show that if you inhibit complex 3's superoxide generation, maybe with compounds like S3-CoL 1.2, IL-10 release goes down.
Less IL-10 means potentially more inflammation or less dampening of it anyway.
Right. And this has even been shown to potentially enhance immunotherapy efficacy in some contexts by reducing that immunosuppressive IL-10 signal. And this IL-10 regulation seems to happen at multiple levels, both in making the IL-10 gene active and in processing the IL-10 protein itself.
Okay, so complex 3 ROS can be anti-inflammatory via IL-10. What about complex 1?
Complex 1-derived ROS, often driven by that reverse electron transport, RET, because of succinate building up in activated macrophages. That's linked more to pro-inflammatory cytokine production, especially IL-1. Now, there's strong correlation and evidence from genetic models, but pinning down the precise causal link directly from RET to inflammation is still an active area. It's complex.
And in an NLRP3 inflammasome, that's a big player in driving inflammation, producing IL-1. How does mtROS fit in there?
Yeah, that's an important point. Some early studies suggested mtROS was critical for just switching on the NLRP3 inflammasome assembly, but more recent genetic evidence suggests it's actually more about controlling the release of IL-1. It seems to regulate a protein called Gasdermin D through oxidation.
Gasdermin D, that forms pores in the cell membrane, right? For pyroptosis?
Exactly. Modifying a specific cysteine on Gasdermin D seems to enhance its ability to form those pores, leading to cell death and the release of inflammatory signals like IL-1. So mtROS influences the output stage.
It really sounds like mitochondria are these central coordinators. And it's not just mtROS doing the talking. Our source highlights TCA cycle metabolites getting in on the action too.
Yes, this is a fascinating convergence. TCA cycle intermediates, like Itaconate and Fumarate, they function as electrophiles. That means they can also directly modify cysteine residues in proteins, much like H2O2 can, creating this overlap in signaling pathways.
Electrophile, okay. Let's take Itaconate first. Sounds like it's a pretty big deal in immunity.
It absolutely is. Itaconate levels shoot up during macrophage activation. We first knew it had antimicrobial properties, but now it's seen as a central immunomodulatory metabolite. It actually inhibits an enzyme called succinate dehydrogenase, acting like an endogenous brake on the electron transport chain itself. And it directly modifies proteins through cysteine alkylation, often dampening inflammation. Synthetic versions, like dimethyl itaconate, can suppress the NLRP3 inflammasome and activate antioxidant pathways like NRF2.
So generally sounds anti-inflammatory, but wait, the source also mentions it can sometimes be pro-inflammatory. How does that work?
Right, this really highlights the importance of context. While it's often anti-inflammatory, itaconate can, maybe paradoxically, enhance type 1 interferon production in certain scenarios by inhibiting an antioxidant enzyme, increasing mitochondrial H2O2. It can also promote inflammatory responses in specific cell types, like certain lung macrophages. So its effects really depend on the cell and the situation. It's not just one thing.
Okay, context is everything. What about fumarate, another TCA metabolite?
Fumarate, like itaconate, is also an electrophile. Its levels are controlled by an enzyme, fumarate hydratase, or FH. If you lose FH function, fumarate builds up. This accumulation can actually trigger a really strong type 1 interferon response, potentially by causing mitochondrial DNA or RNA to leak out. But under normal conditions, endogenous fumarate can also activate that NRF2 antioxidant pathway, helping protect the cell.
And I think I've heard of dimethyl fumarate, DMF, as a drug.
You have. That's a perfect example of the translational significance here. DMF is an FDA-approved drug used for things like multiple sclerosis. It's a stronger electrophile than natural fumarate. It robustly activates NRF2 by modifying its regulator, KIA1, And it also suppresses NF-key signaling, reducing inflammation. It shows how understanding these pathways leads to real treatments.
Amazing. Okay, let's shift gears to adaptive immunity. How do mtROS influence T cells, the sort of precision instruments of our immune system?
Well, just like with macrophages, mtROS are essential signaling molecules in T cell biology too. They influence activation, proliferation, deciding what kind of T cell they become.
Yep.
Their fate decisions. But again, the balance is absolutely critical. And actually, some initial transient ROS increase is needed just to get T cells activated properly.
Okay, what about specific types? Tell me about Th17 cells. They're often involved in autoimmune diseases.
Right. With Th17 cells, it seems excessive ROS actually impairs their differentiation and function. It holds them back. But paradoxically, low basal concentrations of mitochondrial-specific ROS seem to act as positive regulators. They actually promote Th17 differentiation through pathways involving TDF and calcium signaling.
A biphasic relationship. Too much is bad, but a little bit is good.
Exactly. Which makes ROS signaling a potential therapeutic target. Maybe you could fine-tune Th17 responses in diseases where they're overactive.
And what about regulatory T cells, TREGs, the ones that suppress immune responses and keep things in check?
Okay, here's where it gets really interesting, almost counterintuitive. If you reduce overall antioxidant defenses in the cell, leading to elevated total cellular ROS, that actually seems to enhance TREG function and makes them better suppressors.
Higher total ROS helps the suppressors. That seems backward.
It does seem backward. It might help attenuate diseases like colitis. However, if you have high mitochondrial-specific ROS, that seems detrimental to TREGs. It can lead to DNA damage and cell death, which you might see in autoimmunity where TREG function fails.
Wow. So total cell ROS versus mitochondrial ROS have opposite effects on TREGs. The source matters immensely.
It really underscores that point. The specific compartment where the ROS originates appears to dictate the outcome for these critical T cell types.
And CD8 plus T cells, the killer T cells fighting infections and cancer.
For CD8 plus T cells, the story seems to be that lower mitochondrial membrane potential and reduced mtROS production are actually associated with better outcomes. It means improved metabolic fitness, they persist longer, and they have enhanced tumor-killing capacity.
So less mitochondrial ROS is better for the killer cells.
Generally, yes, for their long-term function and effectiveness. Conversely, excessive mtROS, often resulting from chronic antigen stimulation like you see in chronic infections or cancer, that drives T cell exhaustion. It basically wears them out, limiting their ability to fight. This is a huge challenge in cancer immunotherapy, trying to overcome this exhaustion.
So too much mtROS can make CD8 plus T cells tired and ineffective.
Exactly. And some studies have shown that using mitochondrial-targeted antioxidants can actually help restore function to these exhausted CD8 plus T cells, at least partially. It's a complex picture, though. Researchers are using really advanced genetic tools now, like combining mitochondrial complex deficiencies with ways to bypass ROS production to really nail down the specific causal roles of different mtROS sources in both maintaining memory T cells and driving exhaustion.
And ferroptosis, that iron-dependent cell death, it comes up again with T cells.
It does. While CD8 plus T cells can actually induce ferroptosis in cancer cells as a way to kill them, T cells themselves can be vulnerable to it. If T cells lack key antioxidant defenses, particularly the enzyme GPX4, they rapidly undergo ferroptosis, which obviously impairs immunity. This vulnerability isn't just in CD8s, it extends to TREGs and other types like follicular helper T cells too. So understanding these cell-specific sensitivities is vital if we want to develop therapies that trigger ferroptosis in tumors while keeping our immune cells safe or even enhancing them.
This is clearly such a complex and rapidly evolving field. What are some of the biggest challenges researchers are still facing in studying mtROS, and what are the opportunities on the horizon?
Well, a major historical challenge has been the lack of specificity in many ROS detection methods. A lot of the older probes react with multiple oxidants, making it hard to distinguish, say, superoxide from hydrogen peroxide, or to measure ROS just in the mitochondria versus elsewhere. Plus, these molecules are often very short-lived.
Right, trying to measure something specific that disappears almost instantly. How are scientists getting around that now?
They're using a combination of complementary strategies. Things like high-performance liquid chromatography or mass spectrometry give very precise measurements, though not always in real time in a live cell. Then you have genetically encoded fluorescent sensors, like Hyper-7, which can track hydrogen peroxide levels in real time inside living cells. And there are specific chemical probes designed to target mitochondria, like MitoB for H2O2 or MitoNeoD for superoxide.
Okay, so the measurement tools are getting much better, more specific. But how do they move from correlation to causation? How do they prove mtROS causes a specific effect?
That's the critical question, isn't it? Researchers are generating really sophisticated tools now, conditional mouse models, where they can turn on these genetically encoded sensors in specific cell types in vivo. And for perturbing mtROS, there are now highly specific, site-specific mitochondrial ETC inhibitors, like S1QLs and S3QLs, that block ROS production at complex 1 or 3 without just shutting down energy production entirely. Plus, these advanced genetic models using things like alternative oxidase, AOX or NDI1, allow electron transport to continue without generating ROS from specific complexes.
Wow, that allows for really elegant experiments, like removing just the ROS signal while keeping the energy production going.
Exactly. It lets you dissect the causal links much more cleanly. And there are even newer tools emerging, like DTAG systems for targeted protein degradation, or DAO-expressing mice for controlled H2O2 generation in specific tissues. These are revolutionizing our ability to test cause and effect in physiological settings.
And finally, how do they figure out what the ROS is actually doing? Which proteins are being modified?
That's where cysteine proteomics comes in. Techniques like biotin switch labeling or quantitative proteomics using TMT tags help map exactly which cysteine residues on which proteins are being oxidized by H2O2. Once specific cysteines are identified, then researchers use mutagenesis, changing that specific cysteine to confirm if that modification is truly essential for the functional outcome, like immune activation. It's this integrated approach that's really decoding these complex ROS-driven pathways.
This deep dive has, well, it's truly reframed my understanding of mitochondria and reactive oxygen species. They've clearly evolved from just being simple powerhouses to being these incredibly sophisticated signaling centers, intricately weaving into the fabric of our immune responses, both innate and adaptive.
Indeed, it's a paradigm shift. While excessive mtROS definitely drives inflammation and immune cell exhaustion, we now know that controlled generation is absolutely essential for proper immune activation, for differentiation, and even for resolving damage and returning to normal. And the complex interplay with those TCA cycle intermediates, Succinate, Itaconate, Fumarate, just adds another layer of intricate regulation. It highlights this incredibly delicate balance.
The therapeutic potential here seems immense. I mean, from targeting harmful oxidative stress in some diseases to potentially leveraging those beneficial electrophilic metabolites like DMF, which we already use, or maybe itaconate derivatives for inflammatory diseases, autoimmunity, even cancer. It feels like a really fertile ground.
It's an incredibly exciting time in this field, no question. Future research is going to continue defining these precise redox-dependent mechanisms, identifying more key protein targets, and ultimately paving the way for novel strategies to modulate mitochondrial metabolism for therapeutic benefit.
It makes you wonder, doesn't it? What surprising new connections might we discover as we get even more precise in turning these tiny cellular powerhouses and listening to their subtle signals? Hopefully, this deep dive gave you plenty to think about. That's all for this deep dive on the Sand Science Experience. If you enjoyed this conversation, please take a moment to like this video and subscribe to our channel. Your support really helps the Sand Science Experience grow and allows us to bring you more fascinating science. Thank you for joining us.